In this video, we can discuss about the culture media. It's an important topic in the Pharmaceutical Microbiology of third semester B-Pharm. In this, we will discuss about the definition, classification with examples of culture media. Now coming to the definition, culture media can be defined as solid, liquid or semi-solid preparation which are used to grow, isolate, identify, transport or store the microorganism. So from this definition we can understand that the culture media is not only used to grow but also to isolate, identify, transport and store the microorganism. And an ideal culture media should containing all the required nutrients which is essential for that microorganism, and it can be easily selected knowing the natural habitats of the microbial species which will reflect the nutritional requirement, whether it is organotropic or whether it is lithotropic, where it is whatever it is the nutritional habit, what is the classification of that bacteria according to their nutritional habit, it will be easy to select a culture media. Now coming to what are the constituents of a culture media. These are the some essential component that should be present in the culture media. First one is water. Second one is NaCl as well as other electrolyte. Third one is peptone. Peptone, these are protein digest prepared from the animals or vegetable protein by enzymatic reaction. Then fourth one is meat extract, which are prepared fresh from lean meat by hot water extraction. Then yeast extraction from the cells of baker's yeast and Saccharomyces, which are used to enrich the media which containing protein degradation products, carbohydrates, inorganic salt and growth factors. And sometimes we will add blood. Usually defibrinated horse blood will be used. Defibrinated horse blood can be used. Sometimes sera from other animals can be used to enrich the culture media. And seventh one, last one is a carbohydrate derived from the seaweed with an unique property which will melt at 90 degrees Celsius and does not solidify until 40 degrees Celsius. So this is the factor, or this is the constituent which will decide the consistency of an culture media, whether it is solid, liquid or semi-solid. So now we will move to the classification or types of culture media. Culture media is mainly classified into three types based on their consistency, based on their oxygen requirement and based on their constituent. Now first we will discuss about the type of culture media based on their consistency as well as physical state. Culture media is classified into three, that is solid media, liquid media and semi-solid media. The only difference is the physical state as well as the amount of or concentration of agar present in each media. In solid media, that will be solid and it containing 2% of agar, and it is mainly used for colony morphology, pigmentation, hemolysis studies. And example are nutrient agar and blood agar are example for solid media. And second one is liquid media. There is no agar, for it is used for the inoculum preparation, blood culture for the isolation of pathogen from the mixture, example is nutrient broth. There is no agar, so it will be liquid. And third one is semi-solid media. In this about 0.5% agar is used, and which will be semi-solid, and these are used for the study of motile microorganism. Now second classification of culture media is based on the oxygen requirement. It can be classified into aerobic media as well as anaerobic media, whether it is used to help the growth of aerobic organism or inorganic compounds. Now third classification and most important classification of culture media based on nature of constituents or ingredients. It is classified into simple media or basal media, synthetic media or defined media, complex media and special media. In special media, it is again classified into enriched media, enrichment media, selective media, indicator media, differential media, sugar media, and transport media. Coming to the first media, that is simple media or basal media. These are the simplest and most common media in routine diagnostic procedure. Examples are nutrient broth, which consists of peptone, meat extract, sodium chloride, and water. The nutrient broth will be liquid. Now if you are adding that liquid media with 2% agar, it will change to nutrient agar. It will convert it to solid media if it is cool below 40 degrees Celsius. And if concentration of agar is reduced to 0.2 to 0.5, the consistency will be semi-solid. So it can be used for sloppy agar, to obtain sloppy agar, and which are used to study the motile microorganism. Now second media is synthetic media or defined media, in which the all component or constituent of the culture media is well known. We are using a pure chemical substances, and the exact composition of all the ingredient is well known. That's why it is known as defined media. But it is practically not possible. It will be very costly, and it is used in special purposes like for study of metabolic activity or metabolic requirement. For example, for defined media for an photolithotropic autotropy, in this they require photo light energy as energy source, and lithotropic reduced organic compound as electron sources, and carbon dioxide for their carbon sources. So carbon source is carbon dioxide, and nitrogen source is nitrate or ammonia, and sulfate, phosphate, and other minerals for with all non-chemical composition will be used. So that is the second synthetic or defined media. Now coming to the third one, complex media. They containing some ingredient of unknown chemical sample. So there will be cheap because naturally origined compound can be used as a complex media. So it is used when the exact nutritional requirement of species is unknown. We don't know the nature of or nutrient habit of the microorganism. So we are using a general media that is known as complex media, and it can meet the nutritional requirement of various species. We cannot isolate or we cannot separate a single species. We can isolate or we can culture a group of microorganism. Now complex media containing undefined compounds like peptone, meat extract, and yeast extract. It may contain peptones, which are the protein hydrolysis prepared by the partial proteolytic digestion of meat, casein, soya meals, and gelatin, etc. Now beef extract, aqueous extract of lean beef, it containing amino acid, peptide, nucleotide, organic acids, and vitamins and minerals. The yeast extract is aqueous extracts of brevis yeast, and it is a source for vitamin B as well as nitrogen and carbon sources. And the examples of complex media are diptych, soya beet, and Makongi agar. So Makongi agar will become under another example of differential media because according to their use we will classify this culture media in both that one. Special media, these media are used for special purposes, again classified into enriched media, enrichment media, selective, indicator, differential, sugar, and transport media. Now coming to the first one, enriched media. This media we are using to grow the bacteria which need more exacting nutritional requirement. So we will add blood as well as serum or egg in basal media, so it will convert it to enriched media. So the examples are blood agar, chocolate agar, and egg media are example for enriched media. Now coming to the second one, enrichment media. In this we will add a substances which will stimulate the effect of required bacteria, or they will inhibit the effect of unwanted bacteria. So these are used to culture a bacteria which containing more than one bacteria. Examples are tetrathionate broth. In that, tetrathionate will inhibit the coliform bacteria, and it will allow typhoid as well as paratyphoid bacteria to grow. Now another example for enrichment media is selenitive. They will support the growth of dysentery bacillus only. So that is enrichment media. Now next one is selective media. The selective media and enrichment media is only different in their solid state. That means it's also containing a compound that will help the growth of microorganism which is required, and they will inhibit the growth of unwanted bacteria. But these are solid state. So selective media will be solid state, and enrichment media either can be solid, liquid or semi-solid. Now example for selective media is bile salt, basic fusion and crystal violet. They will support the gram negative and they will inhibit the gram positive bacteria. Desoxycholate citrate, they will help for the support the growth of dysentery bacilla and salmonella. Then meconium agar, for it will promote the growth of E. coli and inhibit the growth of gram positive bacteria, and Lowiston Jensen media for Mycobacterium tuberculosis. So that is the selective media. Now coming to next media, indicator media. As the name indicate, they will produce an color change. So media which containing an indicator and which will change the color when bacteria grow in them. So example are sulfate in Wilson and Blair media. In this, if Salmonella typhi is there, they will reduce the sulfate to sulfate and bacteria growing in them will form a black metallic sheen. And potassium tellurate in macleoids media, the potassium tellurate will be converted to metallic tellurium by diphtheria bacillus to produce black colony. So if diphtheria bacillus is there in potassium tellurate in macleoids media, they will produce a black colonies. Now coming to the next media, differential media. Differential media are used to distinguish the various strains of same bacteria with different biochemical characteristics. Example is Meconium agar media, which is used for both selective as well as differential media, but the difference is in selective and differential media we are separating two separate bacteria. Here in differential media we are separating the different strain of same bacteria with different biochemical characteristics. So in Meconium agar, which consists of peptone, lactose, neutral red and taurocolate, which will show a pink color for lactose fermentation bacteria and colorless or pale for non-lactose fermenter bacteria. So that is the difference, differential media. So next one is sugar media, the media which contains sugar, which these are fermentable substances. So they containing either monosaccharide like arabinose, dextrose, mannose, or disaccharide like sucrose, lactose, trisaccharide like raffinose, polysaccharide like starch, inulin, then alcohol like glycerol or sorbitol. So usually sugar media consists of 1% of sugar in peptone water along with the appropriate indicator. So a small tube which is known as Durham tubes is kept inverted in the sugar tube to detect the gas production. So if there is any fermentation, the gas production will be there, so that we can identify the fermentable bacteria by using these sugars. Next one is transport media. As the name indicates, these are the media which are essential for the transporting of media. But in some bacteria only we are using, because that bacteria that are very delicate to handle. So organism like gonococci which will not survive the time taken for the transport of specimen to the laboratory, or there will be a chance for overgrown by some another non-pathogens like dysentery or cholera. So that case we will use this transport media. Example is Stewart media for gonococci and buffered glycerol saline media for enteric bacillus and Carey Blair or Amis media for Salmonella, Shigella and Vibrio. So these are the examples: Stewart media, buffered glycerin saline media and Carey Blair amide media are the example for transport media which are used to handle delicate organs which will not survive during the transport of that specimen. So next one is enumeration media. These are used to count the number of bacteria in the milk and other sample. Just an media to count the bacterial count. And next one is assay media. They are also known as media for special purposes. Profound influence on bacterial cell with respect to the formation of enzyme, toxin, antibiotics and other bioactive products. The pyridoxine deficient growth media for Streptococcus faecalis yield a cell containing large amount of tyrosine and decarboxylase sapoenzyme. So these are used to test the effectiveness of antimicrobial agent, also to assess the effect of disinfectant, antiseptic, cosmetics and preservatives. So that is the assay media which are used to study the effectiveness of antimicrobials, disinfectant, antiseptic, cosmetic, preservatives, etc. So this is an figure which will show antimicrobial resistant zone of inhibition for detection or microbiological assay. Now last one is based on their oxygen requirement is classified to aerobic and anaerobic. All the media which we have discussed is aerobic media. This one is anaerobic media which is used to grow anaerobic microorganism. Example is Robertson cooked media in which nutrient broth with not less than 1 centimeter depth of diced lean hard meat in bottom, and it should be used for used in fresh for recently heated to 100 degrees Celsius to expel the oxygen and then allowed to cool before using the Robertson cooked media. And another example for anaerobic culture media is thioglycolate media. So this is all about the definition, classification with the examples of culture media. And so these are some questions which are seen in the question paper. Define and classify culture media with example. While writing the answer, you should define each culture media and you should give importance to the examples. For each examples, they will be splitting some mark. So you should mention the example for each classification and write short note on culture media. So then write note on material required for the preparation of culture media. What are the constituent of culture media for 5 marks, and what are the difference between some two media. There will be a question like difference between indicator media and selective media, differential media and transport media, like that one question will be there. So these are the regular questions seen in the question papers. Hope it is clear. Thank you for watching this video.